Phosphorylation of glucose. Phosphorylation of glucose is the first step of glycolysis. The reaction is irreversible phosphorylation of glucose to glucose 6-phosphate. Two important features of this step are glucose is trapped as cytosolic glucose 6-phosphate. It means once glucose is phosphorylated to glucose 6-phosphate, it is trapped inside the cell. It cannot come out of the cell membrane. Second feature is glucose is committed to further metabolism in the cell. As a result of phosphorylation of glucose to glucose 6-phosphate, and since glucose 6-phosphate cannot come out of the cell, it has to go to further metabolic pathways. There are many metabolic fates of glucose 6-phosphate, one of which is glycolysis. So this is the reaction showing the phosphorylation of glucose here. Glucose is phosphorylated in the 6th position to form glucose 6-phosphate. The ATP donates the phosphate group to form the glucose 6-phosphate with the formation of ADP, and the enzyme that catalyzes this reaction is hexokinase, and glucokinase is one of the isoform of hexokinase. In fact, hexokinase is the general name of the enzyme, and the specific hexokinase forms the hexokinase 1, whereas glucokinase is also known as hexokinase 4. The phosphorylated glucose is trapped because there are no specific transmembrane carriers for these compounds. Glucose 6-phosphate molecules are too polar to diffuse through the hydrophobic lipid core of the cell membrane. The enzyme, as already discussed, is hexokinase or glucokinase, and these are the isoenzymes of each other. So talking about the hexokinase has got wide tissue distribution and broad substrate specificity. Hexokinase is found in all the tissues where glycolysis takes place. Hexokinase has got broad substrate specificity, meaning it can act upon other hexoses than glucose, like fructose. Hexokinase has got low Km value, that means it has got high affinity for glucose. Similarly, the maximum velocity attained by hexokinase is also low for glucose phosphorylation. The low Km value of hexokinase is significant because it represents high affinity of hexokinase for glucose. As a result of high affinity of hexokinase for glucose, there is an efficient phosphorylation and subsequent metabolism of glucose in the presence of this enzyme. So this is significant, especially when the tissue concentrations of glucose are low. For example, in vital tissues like the nervous tissues. The neurons are in need of glucose. So in the presence of this enzyme, which has got low Km, the neurons can utilize the glucose even when the blood glucose concentration is significantly low. The low maximum velocity of hexokinase is also significant. It represents that hexokinase is easily saturated with glucose. So there is less efficient phosphorylation of sugar, that is glucose, more than a certain threshold. Usually more than the cells can use. So hexokinase can phosphorylate the sugar efficiently only up to a certain point, beyond which the presence of any additional glucose won't increase the rate of conversion of glucose to glucose 6-phosphate. So this graph shows the difference between the enzyme kinetics between hexokinase and glucokinase. So as we can see, the hexokinase follows this type of pattern, wherein we can clearly visualize it has got low Km value and it attains the maximum velocity at this point, which is significantly lower than the glucokinase, having the maximum velocity almost 3 times than that of the hexokinase. And other important difference is the Km value of hexokinase is very low, as shown And by the way, Km is the substrate concentration at which the velocity is half maximal. So at the point where the velocity is half maximal, if we look at the graph, we can find that the substrate concentration for hexokinase is significantly low. So that is the Km value of hexokinase. Whereas in glucokinase, the substrate concentration at which the velocity is half maximal is significantly higher than the hexokinase at this point, where the velocity is half maximal. So if we draw a straight line from here to here and drop it to the x-axis, we can say the point represents the Km value of glucokinase, which is larger as compared to that of the hexokinase. Now talking about the glucokinase, which is also known as hexokinase 4. This enzyme is predominantly present in liver parenchymal cells and pancreatic beta cells. In the liver parenchymal cells, glucokinase facilitates glucose trapping following carbohydrate-rich meal. In the pancreatic beta cells, glucokinase acts as glucose sensor for the insulin secretion. Glucokinase has got high Km and high Vmax values as compared to hexokinase. Now in liver, glucokinase facilitates glucose trapping following carbohydrate-rich meal. What it means is, after intake of meal rich in carbohydrate, the predominant digestion product of carbohydrate being glucose, which is absorbed through the portal circulation, goes first to the liver, and the liver can be said to be flooded with glucose. Now in the liver, glucokinase, because of its high Km and high Vmax values, it traps the glucose inside the hepatocytes and thus prevents glucose from being spilled into the systemic circulation, and thus hyperglycemia during this absorptive phase is prevented to a large extent. Similarly, in the pancreatic beta cells, the glucose sensor, the function of glucokinase as the glucose sensor is important for insulin secretion. Now what happens is, the increase in blood glucose concentration is required for synthesis and secretion of insulin, so to say. Now when blood glucose concentration increases, after a certain threshold, after a certain point is reached, the insulin secretion starts. Now the pancreatic beta cells have got glucokinase, which responds to increased blood glucose concentration by phosphorylating glucose to glucose 6-phosphate, and that step starts the subsequent stages of glycolysis, and that starts the overall process of insulin secretion. Now until that threshold blood glucose concentration is reached, the pancreatic beta cells or the glucokinase present in pancreatic beta cells does not respond to the increased blood glucose until that point, so no insulin is secreted. So insulin secretion and increase in blood glucose concentrations are in tandem only when blood glucose concentration increases a certain threshold. So in that sense, this glucokinase acts as the glucose sensor. The high Km value of glucokinase as compared to hexokinase implies that this enzyme requires a higher glucose concentration for half saturation, and it functions only when intracellular glucose concentration in the hepatocytes is elevated. This is usually significant following consumption of a large carbohydrate-rich meal, where high levels of glucose are delivered to the liver via the portal circulation. The high Vmax of glucokinase is equally significant. This allows the liver to effectively remove a flood of glucose and prevents large amount of glucose from entering the systemic circulation following a carbohydrate-rich meal, and this phenomenon minimizes hyperglycemia during this period.